Hello, my name is Shourav Sharkar and I am a biology tutor, and I hope you are doing well and you are studying well. Today we will cover up the portion that is formation of urine. Okay, that is the main topic, and we are going through the chapter excretory product and their elimination. Okay, and we covered few portions just like what is excretion, removing of metabolic waste from the body, that is known as excretion. Then we covered the excretory products just like urea, then water, and then there is inorganic salts just like potassium, sulfate, then there is you can say sodium chloride. Okay, all of those things are excretory product. There is creatinine also. Okay, then we talked about excretory organ, the major excretory organ, that is kidney, present in the peritoneum cavity on the back side portion, right? On the back side portion there is the kidney, covered by 11th and 12th rib. Okay, there is floating ribs covered by that. And after that we talked about the inner structure of the kidney and other excretory organs just like liver, skin and lungs. Those are other excretory organs. And in case of kidney, what we have learned in the structure, if this is the kidney, okay, kidney has two regions. The outer region that is known as cortex, okay, and the inner region that is known as medulla. Okay, and in the medulla there are renal pyramid. Right? Now in between the cortex and medulla portion, in this portion there will be nephron. And nephron will filtrate the blood and that will collect all the waste product. So from the nephron, all the waste product, that is urine, okay, get collected by minor calyx. Okay, so these are all minor calyx. Then minor calyx will get joined together and that will form major calyx. So this portion is known as minor calyx. When the minor calyx get collected together, that is known as major calyx. And when major calyx will connected together, all the major calyx, that will make pelvis. And from the pelvis portion, it will make ureter. Right? We talked about all of those things. Now today we will talk about the function of the nephron. We talked about the structure of the nephron also, and we will recall the structure of the nephron. And after that we will talk about the formation of urine. What are the steps in case of formation of urine? We will talk about that thing also. So let's start the portion. Everybody open your notebook so you can take notes during the session, and if there is any doubt you can ask me. Okay, Chalo. So what are the steps in case of urine formation? The first step is known as glomerular filtration. That is the first step. Second step, that is reabsorption by tubule. And third, that is tubular secretion. These are the three major steps by which ultimately the urine will form. Okay. Now in case of nephron, there are two major portions in case of nephron. What are those? The first, there is renal capsule. The first part, that is renal capsule, and the second part, that is known as renal tubule. Okay, so let's see what is the renal capsule. We have talked about renal capsule is made up of glomerulus and Barman capsule, right? So renal artery, that will enter and make capillaries. Okay, so the artery, renal artery, that will enter into the kidney. Okay, and renal artery will make afferent arterioles, okay, through which blood will enter. Then it will again bifurcate and that will make glomerulus region, tuft of capillaries, all the capillaries will be there. Then again they will get connected and they will make efferent arteriole. Okay, so through the efferent arteriole all the blood will enter. After getting filtered through the efferent arteriole, all the blood will come out, right? Then after that all the waste product, that could be extra water, glucose. So what could be the waste product? Glucose, water, amino acid. Okay, then there will be urea, inorganic salt. Inorganic salt means there could be sodium, potassium, sulfate, phosphate. Okay, all the inorganic salt. Then there could be creatine. Okay, all of those things will get filtered and enter into the Bowman's capsule. Will there be blood cell? No, there will be no blood cell. Will there be large molecular protein? No, there will not be large molecular protein. Only some small molecule protein will enter. Okay, but large molecular protein and the blood cells, they can't get enter into the Bowman's capsule. Okay, so this process from the plasma, some filtration is happening from the plasma, from the blood. Some portion are getting filtered and entered into the Bowman's capsule. That is known as glomerular filtration. Okay, so let's see this portion. So you can say in the renal capsule portion, glomerular filtration will take place. Glomerular filtration will take place into the renal capsule portion, right? So let's see. So this is afferent arterioles, right? The afferent arterioles will make capillary. Small capillaries will form, and then again those capillaries will bunch together, and that will make efferent arterioles. There will be cup-shaped portion, that is known as Bowman's capsule. So this is Bowman's capsule. And this portion is known as glomerulus. Okay, this portion is known as afferent arteriole. This portion through which blood will come out, efferent arteriole. Okay, now if we see the structure of Bowman's capsule, the outer portion of Bowman's capsule is made up of parietal cells. This is made up of parietal cells. Okay, so outer portion is known as made up of parietal cell. Now the inner portion, in between the connection, the junction point between the glomerulus and the Bowman's capsule, there is a special kind of cells known as, this is the visceral layer. This portion is visceral layer, this is parietal layer or parietal cells, and this portion is visceral portion or visceral layer. Now in the visceral portion, there is a special kind of cell known as podocyte, Okay, so all the molecule, okay, all the filtrate will go through the gaps between the podocytes. These are known as sealed pore. So through the sealed pore, all the filtrate will enter into the Bowman's capsule, and the large molecular, you can say the RBC and the large molecular protein, they can't go through the epithelial cell of glomerulus. They can't go through the gaps between the podocyte. Okay, that's why they will not enter into the filtrate. Okay, only small molecular solutes, okay, glucose, amino acid, then there is inorganic solids, they can enter into the Bowman's capsule portion. Okay, understood? So this is the main thing. Now in each minute, near about 125 ml of blood will get filtered. Okay, so 125 ml per minute the filtration will form. Okay, or you can say 180 liter per day. 180 liter per day. Okay, that high amount of filtration is taking each minute or each day in our body, or you can say in the nephron. So this is known as, this amount is known as glomerular filtration rate. Okay, so glomerular filtration is 125 ml per minute or 180 liter per day. Now what is the main thing? That's why some filtrate are entering into the Bowman's capsule. Is there any kind of pressure? Is there any kind of energy that is being used? So what is the main thing? What is the main reason? Now in this case, for the glomerular filtration, no energy is required. Okay, there are some kind of pressure that will help in filtrate, so now what is that? So let's see. Okay, now here you can see where I have drawn the picture. The diameter of the FR natural is quite large, the diameter of the FR natural is quite small. Okay, why? So if you take a pipe, and in the pipe the one portion is small and another portion is large, what will happen? High amount of water will enter but less amount of water can come out. So what will happen? In that region, high amount of pressure will get generated, right? So just like that, if you take this example, high amount of blood can enter but less amount of blood can come out. Okay, so what will happen? In this portion, high amount of pressure will create, high amount of pressure will be generated, and that pressure will push all the filtrate or all the small particles to enter into this region. The extra water, the small solute, inorganic salt, okay, that will be entered. Okay, so who is giving pressure? Now pressure is generated due to the difference in the diameter, but who is giving pressure? The you can say blood present in the glomerular region. The blood present in the glomerular region, they are pushing, right? They are giving pressure, right? So this is known as glomerular hydrostatic pressure, GHP, glomerular hydrostatic pressure, that is generating in this region. Why glomerular hydrostatic pressure is generated? Due to the large diameter in the afferent arteriole and small diameter in the efferent arteriole. Now only this pressure is there? No, there are two other pressure that is reacting against it, So GHP, who is reacting against it? That is blood colloid osmotic pressure. What is blood colloid osmotic pressure? Now there is the blood, and in the blood there is protein, right? In the blood there are large proteins. That large protein that is present in this region, okay, that will provide some osmotic pressure, and that will prevent the blood, that will prevent the plasma or everything enter into the capsule. Okay, that will generate some pressure against it because they are large. Okay, and that's why some solute can't get entered. Okay, that will prevent, okay, right, to enter into the solute, enter into the Bowman's capsule portion. So that is known as blood colloid osmotic pressure. Now there is another pressure that will be react against the GHP, that is known as capsular hydrostatic pressure. Now all the filtrates that will be collected in the Bowman's capsule portion, okay, they are filled in this region. Now they also prevent the filtrate to enter into the Bowman's capsule. They are present in this region. They are preventing, okay, the filtrate to come from the glomerulus and enter into the Bowman's capsule, right? So they are also reacting against the GHP. Okay, so what is the amount of GHP? That is near about 60 mmHg. Okay, BCOP is near about 32 mm of Hg, and CHP is about 18 mm of Hg. So 18 plus 32, 50. So 60 minus 50, that will be near about 10 mm of Hg. Okay, so depending on only 10 mm of Hg, okay, all the filtrate will enter. Okay, all the filtrate will get collected. Okay, and this thing is known as glomerular filtration pressure. So glomerular filtration pressure, okay, that is 10 mm of Hg, and glomerular filtration rate, that is 125 ml per minute or 180 liter per day. Okay, that is glomerular filtration rate. Okay, understood? Now we talked about glomerular filtration. Okay, how glomerular is filtrating the blood. Then we will talk about the reabsorption of the tubule. Okay, let's see what is reabsorption of the tubule. Okay, here you can see, so from the Bowman's capsule it will start the PCT. Now in the PCT region they have cells, okay, epithelium cells, and if you see the cells in the epithelium, they are just like that. They are cuboidal and they have brass-like structure. Okay, they are brass-like structure. Why they have brass-like structure? So that they can absorb high amount of inorganic salt, water. Okay, so those things can be absorbed in the PCT region. and to increase the surface area, to increase high amount of those things, okay, to absorb high amount of those things in a unit time, they have brass border region, they have brass border structure. We'll talk about that thing. Now that is comes under reabsorption of the tubule, reabsorption of the tubule. This is renal corpuscle, and after that there will be PCT, loop of Henle, then DCT. Okay, and this is part of renal tubule. This is part of renal tubule, and renal tubule will helps in reabsorption. Okay, renal tubule will reabsorb. Chalo, let's see. Okay, so why reabsorption is required? Why reabsorption is necessary? Because glomerular filtration rate is 180 liter per day. So if whatever is filtrated in the glomerular region, if all of those things will be removed as urine, then body will get dehydrated, body will lose the high amount of inorganic salt, body will lose high amount of glucose and everything, then body will become weak. Okay, so in whatever is getting filtrated in the glomerular region, some of them will reabsorb in the tubular region. Let's see. So this portion is renal corpuscle, right? After that, this region is known as PCT, proximal convoluted tubule. Now this U-shaped portion, that is known as loop of Henle, loop of Henle. Okay, PCT, proximal convoluted. Proximal means near to the renal corpuscle, so that's why proximal. Convoluted means they have a convoluted structure. Okay, now in case of loop of Henle, they have two regions: thick descending loop, thick descending loop. This one is thin descending loop, thin descending loop. Thin means that is diameter is small. Okay, then there is thin ascending loop, thin ascending loop. Okay, because it is going towards the upper region, it is going towards the lower, it is going towards the upper region. This portion is known as thick ascending loop, thick ascending loop. Okay, and after that there will be DCT. This region, that is known as DCT. Okay, distal convoluted tubule. Why it is distal? Because it is far from the renal tubule. It is far from the renal tubule. So distal convoluted tubule. Okay, this region is known as junctional tubule. Junctional tubule. Because it is junction in between the renal tubule and collecting duct. This is collecting duct. Okay, so collecting duct will enter into the portion of renal pelvis, then from renal pelvis to ureter. Okay, all thing will be transferred. Okay, now in the PCT region, high amount of absorption will take place. So whatever entered in as a filtrate, okay, from that filtrate, whichever is necessary, some salt, some amino acid, then glucose, that will be reabsorbed in the PCT portion. So in this PCT portion, near about 65% water will get reabsorbed, so that body can't be dehydrated, body would not be dehydrated. Okay, so in the PCT portion that will happen. Okay, some amount of glucose, amino acid, vitamin, okay, all of these things will get absorbed. So water will be get absorbed by the process of osmosis. All of these three will get absorbed as the process of you can say active transport. By the process of active transport, they will get absorbed. Okay, now in this portion, in the loop of Henle portion, okay, in the loop of Henle portion, the water only get absorbed. The water only get absorbed. Okay, not the solute. Okay, not the inorganic salt or anything, but the water will get absorbed. Now think about the portion. From a solution, you are absorbing the water only, and solutes are there. Okay, so what will happen? The concentration of the solution will get increased, right? So in the tip of the loop of Henle, the concentration of the filtrate, now it is not urine, it is filtrate. Okay, or you can say primary urine. So in this region, the concentration will be high. So concentration will be high because only water is absorbed, solute is there. Okay, concentration will be high. Concentration will be high for filtrate or you can say primary urine. Now in the thick descending portion, or you can say the ascending portion of loop of Henle, in that case water will not absorb. Now solute will absorb. What will happen? Solute will absorb. Okay, water will not get absorbed in this region. And when the water solute will get absorbed by the process of active transport, then so from a solution, okay, you are taking all the solute. What will happen? Concentration will again decrease. So concentration was high because till now we are absorbing all the water. Now we are taking all the solute. So what will happen? Concentration will again become diluted, right? So concentration will become diluted. In the DCT portion, the solute will absorb the sodium, potassium, phosphate, okay, all of those things absorb. And in this region, the absorption of sodium is depending upon a hormone, okay, that is known as aldosterone. Okay, aldosterone that is released by adrenal cortex. So if this is the kidney, on the upper portion of the kidney there is a triangular shaped organ that is known as adrenal cortex, and from the address that is known as adrenal gland, and from the adrenal gland they will release a hormone known as aldosterone. Okay, and aldosterone will absorb sodium. Okay, if there is low amount of aldosterone, the sodium will not be absorbed, it will be released by the urine. Okay, that is the first. Now in this region water will get absorbed. But in that case there is another hormone that will help in absorption, that is known as ADH. Antidiuretic hormone. Okay, that is released by pituitary. Pituitary present in the head, that is the endocrine gland, and that will release the ADH to absorb water. Now if the ADH is not released, if the water is not absorbed, what will happen? High amount of water will be released from the urine. There will be frequent urination. As the water is not absorbed, high amount of urine will be produced and high amount of urine will be released from the body. Okay, and that is known as, that frequent urination is known as diabetes. You can say diabetes insipidus. Okay, that is known as diabetes insipidus. There is two diabetes: diabetes mellitus, diabetes insipidus. Diabetes mellitus is due to high blood glucose level, diabetes mellitus, and diabetes insipidus is due to frequent urination. Okay, so that is the second portion, due to ADH. Okay, so reabsorption of the tubule, that will be up to this portion. Now in the collecting duct portion, again some portions will be absorbed, that is hydrogen ion, potassium ion, okay, urea, small amount of urea, that will also get absorbed. Okay, so these things will get absorbed again in the collecting duct portion. Okay, then it will form the urine. Okay, so in the collecting duct portion, you can say the first urine will be formed. Up to this portion, it is known as filtrate or it is known as primary urine. Okay, understood? That is reabsorption of tubule. Okay, now tubular secretion, we will talk about tubular secretion. If there is any doubt anyone, you can ask me. Okay, so let's see. Okay, now tubular secretion. So for tubular secretion, a important structure is there. Okay, this is afferent arteriole, then there is glomerulus. Okay, now there will be efferent arteriole, right? There will be Bowman's capsule. Now this efferent arteriole, that will form another structure of capillaries, that will make another capillary structure. Okay, that will make a network like structure. Okay, understood? Surrounding the DCT and PCT, So what is happening? Why they are making a net like structure surrounding the DCT? Because if you see, if this is the DCT, This is the capillary that is made by the efferent arteriole. These capillaries are known as peritubular capillary. This capillary is known as peritubular capillary. Peritubular capillary. Okay. Now peritubular capillary, what is happening? So when the blood will go through the glomerular region, okay, some of the solute, some of the medicine, some ions, okay, that will be not filtered. There is a chance they will not get filtered due to those blood colloid osmotic pressure, due to capsular hydrostatic pressure, okay, and they will enter into the blood. They will enter into the blood, okay. They will go through the blood without filtration. Now the peritubular capillary, through which the plasma is going, okay, and that capillary will end up into making renal vein, okay. So before blood entering into the renal vein by carrying all of those toxic products, there is a second chance, or there is a second chance that from the plasma it can enter into the DCT again. How? So from the peritubular capillary, okay, peritubular capillary, the ions, extra ions, that is hydrogen ion, then bicarbonate ion, then there is the some medicines, okay, they will enter from the peritubular capillary, mainly from the plasma or blood, you can say blood, okay, to the filtrate, and through the filtrate it will enter into the collecting duct and released by the urine, okay. So peritubular capillary releasing or secreting, tubular secretion. So tubule is secreting the toxic product, okay, or the waste product in the distal convoluted tubule, okay, so that they can maintain the proper plasma concentration, or they can maintain the texture of the plasma, the composition of the plasma, okay. Why they are not filtrated into this region? Due to some opposite pressure, there is a chance that some of the molecule can get filtrated, okay. So they will filtrated into the DCT region, okay, from the peritubular capillary they will enter, okay. So that is tubular secretion. So this is the whole process by which urine will get formed into the nephron region, okay. So this is the whole portion. If there is any doubt, you can ask me, okay. So go through the portion, read the portion, okay, and read the portion what you have made notes. So see you in the next class, okay. So take care, bye bye.